Guys, today we're going to be talking about the Nuremberg and Mathai experiment, which was conducted to figure out what amino acids are specified by codons composed of only one type of base. First, they created a cell-free system using ruptured E. coli cells that contained enzymes and an energy system. Then they added the homopolymer poly-U, which is made up of multiple uracil nucleotides. They also added a radioactive amino acid, which is represented by the star, and then 19 other unlabeled amino acids. And all of these were added to the test tube, as you can see here at the bottom. The test tube was then placed in an incubation system set to a temperature of 37 degrees Celsius. After incubation, we can see that translation in the proteins occurred, which caused a precipitate to be formed at the bottom of the test tube. Next, they filtered out the precipitate protein using a filtration device in order to separate it from the rest of the mixture. This protein would later be checked by them in order to see if there was any radioactivity. They found that the radioactive labeled protein led to newly synthesized phenylalanine, meaning that 3 uracils coded for phenylalanine. Finally, they repeated the experiment in 20 test tubes, with each one containing a different labeled amino acid. Again, they found that the test tube with radioactive protein contained newly synthesized proteins with the amino acid specified by the homopolymer, and this concluded that there was in fact a genetic code. The next experiment Nuremberg conducted that led to the completion of the genetic code was with later, in 1964. New technology allowed for all codons to be made in vitro. They began by synthesizing artificial mRNA codons. These codons were then added to a test tube. Amino acylated tRNAs were also added to the test tube, as well as ribosomes. The mixture was then put through a filter that traps the ribosomes at the top and allowed the tRNAs that weren't bound by ribosomes to pass through the filter. So here we can see the unbound tRNAs at the bottom and the tRNAs that were bound to ribosomes and the mRNAs at the top of the filtration system. As an example of how these findings led to the genetic code, when an mRNA with the code AAA was added, the tRNAs on the filter were bound to lysine.